Hello, welcome to this week's MOOC on constraining neuronal models with experimental data. My name is Felix Schürmann. I'm a professor at EPFL working at the Bluebrain project, where for the last couple of years we've been working on the reconstruction and simulation of realistic neuronal tissue. In today's part of the MOOC, we will recap the different mathematical levels of abstraction that are useful to describe neuronal behavior. And I'll make the point that the multi-compartment Hodgkin-Huxley formalism that were described in other parts of the MOOC are a good starting point for realistic neuron models. We'll be talking about experimental datasets that can help us to parameterize these models and the challenges that occur in that process. That should keep us busy for this part of the MOOC. So if you have a neuron in mind which you want to model, and in this case it's a layer 5 pyramidal cell of a young rat, and what we want to model is, for example, how would a somatic recording to a step current injection look like, how an action potential is generated, or something similar, or other electrical behavior. You have different possibilities to try to cast it into a mathematical model. And it starts with simply modeling the cell body, the passive electrical properties of that, and use a template mechanism activated after crossing a certain threshold to initiate an action potential. Or you can go to the Hodgkin-Huxley formalism that extends these types of modeling with active conductances for sodium and potassium currents and allows to describe the action potential as an emergent property of the electrical system. Or you can extend that further and say, I'm not only modeling a single compartment, but I would like to have the formalism to describe the dendritic tree and the cable equation of that. And this was described, for example, by Idan with the RAL equations for combining the RAL equations with the Hodgkin-Huxley equations to have a full biophysical model of a spatially extended neuron with an action potential generated by the biophysically correct conductances. But it doesn't stop there necessarily. You can imagine that you can go into even more detail and sort of include biochemical reactions, electrodiffusion, and ligand-gated cascades or other things, and then you would have to change the mathematical formalism again and would have to characterize yet other properties that lead to, for example, something like an action potential. For the purpose of this MOOC, we will stick with the multi-compartment Hodgkin-Huxley formalism, essentially for two reasons. On the one hand, computationally speaking, it is still the simplest mechanism that can capture spatially extended features of a neuron and treat the electrical behavior as an emergent property. At the same time, it has enough hooks in terms of the individual compartments it's modeling and the different conductances you can add, that it is very amenable to include new biophysical detail. And it's exactly these two properties that it's computationally still tractable and yet, on the other hand, allows to include a lot of biophysical detail that makes this formalism very interesting for realistic neuron modeling. When spanning the timeline from the original experiments and modeling of Hodgkin and Huxley, then the theory of RAL, to today, there are an important number of milestones for this type of modeling, which I think I briefly would like to capture. On the one hand, in the end of the 70s, Roger Traub and Linas started to extend the level of detail of the cable equations that were solved up until that date. But note the type of geometries you see here are still very much motivated by making the geometries more realistic, but still as simple as possible. So here you still see the 2/3 power law, for example, that simplifies certain mathematical equations. This is a hippocampal motor neuron. And at the time, this was computed on a mid-frame computer. In the same year, for a different morphology, here we're talking now about Purkinje cell of the frog, these simplifications no longer held. So here the dendritic tree doesn't show these symmetries, and therefore this model has about 60 compartments and does treat these compartments in their detail, having to solve the full compartmental equations in Hodgkin and Huxley. It took another couple of years before really the morphologies went really detailed. And so this is now work by Eric de Schutter and Jim Bauer on the Purkinje cell. And here we now have a full dendritic reconstruction of the Purkinje cell tree. And the neuron model they did here had 1,600 compartments, 10 different channel types with differential channel densities, recreating multiple different parts of observed behavior of that neuron. And lastly, yet a couple of years later, Meinen and Sosnowski had modeled a pyramidal cell where essentially they could show that the change in somatic firing you see which is of course, for example, mediated through different channels or different channel densities, but that alone a dendritic tree can influence the somatic firing pattern. So what we see here in these last decades of detailed modeling, that on the one hand, the computational available power from mid-frame to personal computers to more powerful computers, thanks to Moore's law, actually allowed to integrate much more detail. And at the same time, of course, that this more computational detail necessitates having more experimental data available, but at the same time allowing to integrate more experimental data. So when we're talking about realistic neuron modeling today and constraining neuron models with experimental data, we have essentially the computational power available, and we can think about primarily which data sets we can use to constrain the models. But the starting point is still very much the same. You want to have a certain morphology for the neuron of interest. You have possibly certain electrical behavior that you've recorded, suprathreshold, subthreshold, from the soma, from the dendrite. And in the end, you'll have to find how do I constrain all the parameters that are exposed in a multi-compartment Hodgkin-Huxley model. So we're talking about the topology, the passive properties of the cable, the active conductancies in the different parts of the tree, their behavior, and sort of what's the how many of those conductancies you have where. We will now go through looking through these different data sets and what are the things you might want to keep in mind when using them. We start with the morphologies, and first of all, a very good source to get a morphology reconstructed is to go to neuromorpho.org. Now we pick here again our favorite neuron, which is the layer 5 pyramidal cell, and the neuron looks beautiful if you look at it in this XY coordinate frame. But what you should always do, that from seeing a beautiful neuron on a screen or on a paper, that is not the same as saying that the morphology has all the parameters needed to constrain your model. And this is one example what could go wrong. So if you turn this neuron around by 90 degrees, you find suddenly that there's almost half of the neuron of the dendritic tree and the axon missing. Why is that? Well, this particular neuron was recorded and stained in slice, and it was at a certain depth. At some point the slice ended, and that meant that certain parts of the neuron are shaved off. You don't see that in this perspective, but you see it when you turn the neuron around. So keep in mind that depending on what is the recording, staining, and reconstruction technique, you have to check whether actually you have all the data in your experimental data to constrain a computational model, because it matters whether that part of the tree is there or not. Now, there are other systematic artifacts to look out for, that some staining and fixation methods lead to shrinkage or sort of crumbling, and that leads to wiggly branches, which again, or sort of other artifacts that could have computational implications in your model. And you always want to make sure that sort of some other things, like for example axons, are they stained in the first place? Were they reconstructed? Are they complete? All these things suddenly matter, because essentially an axon which is not there will not parameterize the cable in your model. Unfortunately, the story of morphologies does not end there, and there are a number of other little things that can keep you off track when using experimental data to constrain your model. It can start with that at the reconstruction process, the diameter wasn't explicitly given. It's still, the software might connect 2 points even though they have 0 diameter with a line, but electrically that is not a good parameter to have in your cable equation model, to have a diameter of 0 in a certain compartment. You might see that due to the way that these reconstructions happen typically from X, Y, and then the Z is something that's done through scrolling through the stack of the slice, that when reconstructions get started and resumed, and restart it, that's sort of not always the reconstructions are fully aligned. Again, in the XY plane, you won't see these types of artifacts. There are sometimes things like reverse order reconstructions, that sort of things were traced inside out and not starting from the cell body. Think again, when you look at the topology, of course it matters tremendously where these different compartments are connected with each other. At the same time, sometimes some things are not connected at all, or spines get reconstructed as single segment sections, or you might find that certain parts of the morphology get reconstructed several times. Again, on a visualization of such a morphology, that is not an issue. For electrical modeling, all these parameters lead to different properties of your cell model. So the take-home message here simply is that using experimental data for realistic neuromodeling is crucial, but you have to make sure that the experimental data set sort of has the accuracy and the quality so that it gives you a good electrical passive frame for your modeling. Moving to ion channels. So as per the work of Hodgkin and Huxley, we know that sodium and potassium currents are the key to generate an action potential. So you could say, well, I want my cell to fire, I put sodium and potassium currents, that's it. But of course, the story is not as simple, and sort of there are many more details to be possibly considered when you're looking at your particular neuron in question. And first off, these currents are mediated by different ion channels. So there's not just one sodium and one potassium channel type, but they're, as a matter of fact, different ones. And these sodium-potassium channels shape a lot sort of how the neuron depolarize, how the action potential gets initiated, how the after hyperpolarization happens. And of course, there are additional ions, there are calcium, chloride, and other ions that shape additional parts of the neuron. It can be the onset or the spike or the persistent firing behavior of your neuron. So again, if you have a neuron that, for example, is a delayed spiker or a burst spiker or something else, you will have to add additional detail that is not in the original sodium and potassium parameters of Hodgkin and Huxley. Now, as a matter of fact, there are about 350 ion channel types known in mammals, and most of them seem to be expressed in the nervous system. They roughly categorize into three different groups. There are about 150 voltage-gated channels, there are about 90 ligand-gated channels, and there's the third group, others, aquaporins or chloride channels. And so in principle, when you're looking at your neuron, you're looking for data, you're looking at having to identify which of these 350 channels might be in your neuron. But before we go there, first of all, what do we know about these channels? And here you see an overview of the voltage-gated ion channel family. And in this case, you can, of course, classify them in different ways. You can classify them by ion, for example. But here they're classified by, on the one hand, their pores and then the similarity in the amino acids. And you see that sometimes some channels have 4 domains. And then sort of that makes them more similar, even though they're letting through different ions, than, for example, calcium channels to other calcium channels. Or, on the other hand, sort of different potassium channels to other potassium channels. So the take-home message of that is that here there's a wealth of ion channel experimental data that might be useful for your parameterizing your neuron model, but of course you have to know whether sort of which exact channels you might have in your neuron, and then the second point, what is known about that channel. And then you can use the data to characterize the particular activation and inactivation parameters in your ion channels that sort of you put into your Hodgkin-Huxley model. Now, of course, that's exactly the question. Which of these channels are in my neuron? And there are different ways of finding, going back to experimental data to help you with that decision. There are pharmacological methods. You can do antibody staining. You can do single-cell multiplex reverse transcription PCR, or nowadays you can also try to do single-cell transcriptome. It is all about asking what in a given cell, which genetically identified ion channels you might have. And this is a diagram of a data set which was done through multiplex reverse transcription PCR. So essentially there's some cytoplasm extracted from a cell, and then you have different genetic probes to sort of see whether you can fish mRNA sequences for particular ion channel genes out of a microplasm. Now, of course, having mRNA is not the same to say that you have the protein expressed. So these last two methods, in a way, give you an indication that there's the genetic machinery has been activated to produce a certain ion channel, but it doesn't mean exactly that you know for sure that the ion channel is there. But there is a high correlation. But that being said, sort of from these data sets, you can see that even though these two cell types seem to have somewhat similar sustained firing behavior, they differ in the onset. So this is delayed, this is burst spiking. What you can see is that for the ion channels visible here, you might see a completely differential expression of these channels leading to these type of firing behaviors. So these are invaluable data sets to, out of the jungle of ion channels, choose which ones of those might be present in your neuron, then helping you to select which channels you have to model in the first place and where you can look for parameters for your model. Next, when you now know which channels you have to consider, you will have to ask yourself the question, do you know the different parameters like activation and inactivation parameters for the channels you want to put into your model? And a good source for channel models is ModelDB, which is hosted at Yale University. This is the URL. And that's a very important resource because there they track published models of channels, cells, and networks. And it's sort of allows you to go back and sort of find out which parameters were used in a different model. So in principle, once you know your neuron model and the channels, you might be able to go to that resource and ask the question, can I take the parameters from that channel model? Now in practice, the problem is a little more complicated, which prompted an activity which you see here, this part of the screen, which is the IC Genealogy project, which is hosted at Oxford. What they did is they went to the ModelDB and extracted every single ion channel model, and you now have different ways of displaying those ion channel models. You can display them by channel family or current family, but you can also ask the question sort of which model was derived from which other model and how similar are these models. So as a matter of fact, this is a different way of looking at the jungle of ion channels, but namely from the model perspective. What this really means is that different modelers for different models might have taken another modeler's ion channel and then changed it. To simply say, look, here the equations were modified from Traub, and we, for example, changed the conductance of the potassium channel, and the reason why they were changing the channel properties in the model was likely that they wanted to have their cell or their network behave in a certain way. So as a matter of fact, the channel parameters were treated as a modifiable or free parameter, so that in order to get a later or higher order behavior, you could adjust channel models. But what that really means is that you depart from the biophysically characterized channel as, for example, done by an experiment. So, of course, there's no criticism in this, because in the end, you have to ask yourself the question, how good is the experimental data in the first place? Does it have enough quantitative value to create specific ion channel models, or sort of were there approximations in the first place? But what it really means is that it is not necessarily given that models of channels and models of cells use the experimental quantitative data for modeling the channels, but that there was, due to also the absence of data, a large period of time when models sort of had to guess channel parameters. Now, the good news is that this may change with time. And here I would like to briefly present an endeavor which is called Channel on Channelpedia. And here you can go to Channelpedia, where you will find a good resource of ion channels expressed in the brain, and as well as data sets and increasingly more models. And this project has been set out to create cell lines that have genetically identified individual channel types expressed, and then systematically characterize these channels under different temperatures. So it is actually trying to systematically obtain the kinetics of all these genetically identified ion channels. So once this is complete, you will no longer have to go to a colleague's channel of a model and sort of modify it for your means, but you will be able to sort of create channel models which are backed by data, effectively reducing the parameters that are at your disposal when sort of creating cell models, making the problem simpler because you have less parameters to find. So this is a typical example for how increasing experimental work helps you to sort of extract more and more parameters for your realistic neuron models. Now, you can ask yourself the question, sort of, why wasn't that done before? Why are we still today talking about that effort? And the secret lies in a way of how interesting ion channels are in the first place. Some of these channels were found to be implicated in certain diseases, and then a lot of studies have been made about the channel, but some of these channels are not interesting for disease, they're exotic, and we know very little for it. And also, the genetic characterization wasn't known since the beginning of ion channel works, so especially Hodgkin and Huxley. So we knew about currents, but we didn't know about specific ion channels. So as a matter of fact, it is a particular effort to systematically find and characterize those channels, and we think that has very good value for realistic neuron modeling. Lastly, once we have morphology, channels, the channel kinetics, we still have to answer the question as to where to put them and how many of those channels. And that is a complicated story. Some neurons, of course, are simple, so they are small in the soma and just small dendritic trees, like the granule cell, where you can actually, through electrical recordings, infer the maximum conductance of channels. But other neurons with an extended dendritic tree, it is very difficult to infer from a somatic recording what could be the channel distribution in the dendrites or the channel conductances. So as a matter of fact, through staining, pharmacology, recordings, we can piece together some information about that. Sort of some channels are only in the soma, or some channels are more in the apical dendrite, but the matter of the fact, the picture remains dirty. So as it stands today, the question of will we able to quantitatively extract channel distributions and maximum conductances, I think remains an open question, and we will take and talk in other parts of the MOOC sort of what we can do to mitigate that challenge. Let me summarize today's course on constraining neural models with experimental data. So when you're setting out to build realistic models for a specific neuron, you're faced with having to identify multiple parameters: neuron's topology, diameters, which type of ion channels, ion channel kinetics, maximum conductances, and so on. We think that in order to make these models meaningful and the problem achievable, it is very important to use as much biological data, experimental data, to constrain these parameters. So essentially make the problem simpler by reducing the number of free parameters. We think in particular that multi-compartment Hodgkin-Huxley formalism is a good formalism to allow you to integrate experimental data. It has the right hooks to sort of include specific parameters for an ion channel or specific densities in a certain compartment. So in that sense, the multi-compartment Hodgkin-Huxley formalism is not only a data-driven formalism, it is data-ready. It makes it easy to absorb data. That being said, there are particular parameters that are remain more experimentally inaccessible than others, and that might make it necessary to employ other methods to sort of help find and constrain these parameters, which we will be talking in other parts of the MOOC. Thank you very much for your attention, and hope to see you in the next part of the MOOC.